So, chapter 3 is about cells, and every living organism is made of cells. So, animal cells, like humans, like other animals that you see, are different than plant cells, but everything that lives is made of cells. Your skin is different from your muscle because skin cells are different from muscle cells. Cells specialize by changing themselves through differentiation. Cells are like factories that produce different components. So here we have a stylized version of a cell. No cell has all these parts, okay? They all have some of these parts, but you have the cell membrane on the outside, and you have the cytoskeleton of the cell. These purple pieces that look like canals are ribosomes. They're the little factories that get produced. You have the mitochondrion; those are the energy machines. You have the centrioles up here, and the lysosomes, the little tiny dots up there. And then you have the flagellum. You have the nucleus of the cell where the protons and the electrons are. And then you have rough endoplasmic and the smooth endoplasmic. If you scroll up, you'll see. And then you have the Golgi apparatus and the smooth endoplasmic reticulum. So between the smooth endoplasmic reticulum and the rough endoplasmic reticulum, that's like the highway through the cell. And then you have cytosol, and these are animal cells because they only have one wall. Plant cells have a double wall usually. So the cell membrane is made up of, it separates the living cell from its non-living components, and it is made of largely phospholipids. Again, we've got the hydrophilic head, the hydrophobic tail. You've got the fatty acid chain. You've got glycerol up here, phosphates. And then, of course, the polar group, the hydrophilic head, where the polar water molecules are. Okay. The cell membrane is selectively permeable. The cell membrane, the plasma membrane, controls the movement into and out of the cell. Oxygen, carbon dioxide, and other small nonpolar molecules, and some water molecules go through, and glucose and other large polar water-soluble molecules can't get through. So this means that certain selectively permeable means that some things are able to pass through the membrane. Think of like cheesecloth or a strainer, right? If certain things are too big, they're not going to be able to pass. So things like glucose sodium, potassium, calcium, chlorine, hydrogen, they're not going to be able to get through the membrane. Things like oxygen, carbon dioxide, some water molecules will be able to pass through the membrane. Okay, so then we have the fluid mosaic model of the cell membrane. A diversity of proteins float about in that phospholipid bilayer. We're going to talk more about the proteins that are in your phospholipid bilayer later. I guess he just wanted to present it, or she just wanted to present this, but not in a great deal of detail right now. So we have the cytoplasm, which is inside the cell, versus the nucleoplasm. So the cytoplasm is sort of the liquid that's inside the cell itself. And inside the cell, we have the nucleus. The nucleus has stuff inside of it too. So the cytoplasm versus the nucleoplasm. So cytoplasm is the semi-fluid gel-like matrix between the cell membrane and the nucleus. Organelles are suspended in the cytoplasm. So organelles like the centrioles, the ribosomes, they're the mitochondria, they're in the cytoplasm. Nucleoplasm is the semi-fluid gel-like matrix within the nuclear membrane. Now, what does the nucleoplasm contain? So this is inside the nucleus. What does the nucleus contain? Well, it has the nuclear membrane, has the nucleoplasm. It has chromatin, which is sort of the precursor of genetic material, DNA, genes, proteins, and it has chromosomes that allow the cell to reproduce. And if they happen to be sex cells or gametes, then they're the ones that determine whether the organism is going, if it's an animal, it's going to be male or female. Okay, so the nucleus is the control center. It keeps the DNA molecules from the metabolic machinery of the cytoplasm. The nucleus has functions. It acts as the genetic control center. It directs cellular activity, and it stores hereditary information. The nucleolus is the center of the nucleus where ribosomes are assembled. So there's a center of the nucleus called the nucleolus, like a smaller nucleus, where the ribosomes get produced. So it makes it easier to organize DNA and copy it, and it contains the chromosomes. The nucleus contains the chromosomes. Okay. The endoplasmic reticulum starts at the nuclear membrane and extends throughout the cytoplasm. The rough ER, the rough endoplasmic reticulum, is covered with ribosomes, and it builds special proteins, which makes sense because ribosomes are the protein factories of your cells. Smooth ER doesn't have any ribosomes. That's why it's smooth ER, because it doesn't contain ribosomes. It's not covered with anything, but it builds lipids and it handles toxins. Ribosomes translate the DNA blueprint into proteins. Ribosomes make protein by a process called translation, and they don't have a membrane. So down here, you have the pictures of what we saw before the cell. You also have, this is a blown up picture of it, and then this is the protein synthesis. You have the ribosome, the plant factory. You have the amino acid coming out, and remember, amino acids are the building blocks of proteins. So this red piece here is the protein chain coming out. Okay, then we have Golgi bodies. Golgi bodies do the packing and the shipping. So they put the finishing touches on proteins that arrive from the rough ER, from the ribosomes in the rough ER, and the lipids that arrive from the smooth ER. Packages are finished material for shipment to the final destination, and the material leaves and arrives in vesicles, seminal vesicles, usually something. There's different kinds of vesicles that we're going to hear about, but those take place in the Golgi bodies. Then we have the mitochondria. As I mentioned, they're the power factories of the cell. They produce energy, ATP. They break down. They produce and break down ATPs. They're the site of cellular respiration, and their membranes form two distinct compartments. Lysosomes do the cleanup. They're the janitorial department. They are membranous sacs that contain digestive enzymes. So organelles get old. Equipment gets old. You have to repair it. You have to recycle it. In some cases, you have to junk it. Lysosomes do the cellular cleanup in this area. They keep the cell from getting crowded by removing waste and debris. They also, let me see, they also digest food or bacteria that is phagocytized by animal cells. So when you have bacteria that cells in our bodies get rid of, when the white blood cells engulf them and they eat them, phagocytosis is basically a word that means cell eating. They eat the bad cells. Lysosomes digest that. Lysosomes absorb that so that it doesn't stay in the body. The cytoskeleton is the framework, the walkways, and the conveyor belts. They're present in all eukaryotic animal cells. They're not bound by a membrane. They help the cell to maintain structure and internal organization, and they allow the organelles, the smaller organs within the cells, to move, and in some cases, they aid with cell motility, the cell's ability to move in the first place. Now here you have a picture of different ways that cells or bacteria can move. Sometimes you have organisms that move with flagella, which are basically long tail-like structures, flagella. So we've got this long tail, and it moves by swishing its tail around. Got the head up here, and the neck, and then the body and the tail. So it moves by moving its tail around, flagella. And cilia are hair-like structures. So where you find cilia are like in your lungs, you have bronchial cilia. If you get bronchitis, if you've ever had that, then that usually your cilia get inflamed. Your bronchial tubes that are lined with cilia, your bronchial tubes get narrower because they get obstructed with mucus, and it makes it hard for your cilia to filter things. They're small, like hair-like structures. They're not hair, but they look like hair. They move like hair moves. Cilia. Okay, go ahead. Okay, again, there is no cell that looks like this, but this is a stylized picture of the cell with all of the pieces showing for you here. There's no cell with all of that. Okay, but all of those pieces are showing. Okay. So we're working on chapter 3. All right, let's look at chapter 3 and see what we've got. All right. As in cytoplasm, cyto refers to the cell. CELL. Which of the following is the prefix endo? Endo means like the endoplasmic reticulum, right? You can review it if you want to review it. Okay, I see the picture. Okay. Okay, so here's some of the prefixes that they're asking us. Cytoplasm, fewer pills, cyto, cyte means cell. So you're going to look at the ones in brackets. Endo is within, as in endoplasmic reticulum. Hyper means above. Hypo means below. Inter means between. And iso means equal. So endo is within. Within. The prefix blank means above, as well as greater or higher. That's hyper, I think. Go ahead. You can read it. Hyper. Hyper. Hyper means above. Okay, which one means below? Okay. The smallest unit of organisms like humans that are considered to be alive is a cell, tissue, molecule, chemical, or atom. The smallest unit.
Maybe cell.
That's not the smallest. Go look and see. It could be if it's alive, but go look and see at the resources what they tell you. Cells are the smallest units, and the smallest units in the cells are atoms. So cell is what they wanted. You were right, and I was wrong here. Yeah, good. Cell is correct. Okay, next question. The prefix blank, okay, we did this before. Which one means cell?
Maybe the second one.
Yeah, it just has the O missing. Sometimes it has the O, sometimes it doesn't, but that means cell, yes. Good. All human cells have a phospholipid bilayer that makes up the cell blank. Cells differ greatly in size, shape, content, and function. Therefore, describing a typical cell is challenging. The cell shown in figure 3.2 and described in this chapter is a composite cell that includes the usual cell structures. In a living organism, any given cell has most, but perhaps not all of these structures, and cells have differing numbers of some of them. Okay, but that's not the answer to the question yet. Keep going. Under the okay, cell membrane, and that includes the nucleus, cytoplasm, organelles, which are seen. Okay, keep going. The cell membrane, also called the plasma membrane, is more than a complex simple boundary holding the cellular contents. It is an actively functioning part of the living material. The cell membrane regulates movement of substances in and out of the cell and is the site of much biological activity. Many of a cell's actions that enable it to survive and interact with other cells use a molecular communication process called signal transduction. A series of molecules that are part of the cell's membrane form pathways that detect signals from outside the cell and transmit them inward, where yet other molecules orchestrate the cell's response. The cell membrane also helps cells attach to certain other cells, which is important in forming tissues. A cell membrane is thin and flexible and is composed mainly of lipids and proteins with a few carbohydrates. The cell membrane maintains the structural integrity of the cell and is selectively permeable or semi-permeable. Okay, I think we're getting warmer. Keep going. Which means only certain substances can enter or leave a cell. Its basic framework is a double layer or bilayer of phospholipid molecules. Each phospholipid, okay. Each phospholipid molecule includes a phosphate group, 2 fatty acids bound to a glycerol molecule. We talked about that last time. Chemical, the water-soluble phosphate heads are hydrophilic and form the intracellular and extracellular surfaces of the membrane. The water-insoluble fatty acid tails are hydrophobic and make up the interior of the membrane. Okay, what else does it say about the phospholipid bilayer? Does it say anything else? Okay. Keep going. I want to see if it says, okay, I'm not sure what it wants. Go back to the question. All human cells have a, I think all they want there is membrane. All human cells have a phospholipid bilayer that makes up the cell membrane. I was thinking they wanted to know more about the membrane. Maybe there's another question about the, go ahead. Yeah, I was thinking they were asking us about the cell membrane, but not yet. Okay. Which one means within? Do you remember?
Endo.
Endo, correct. Uh-huh. Good. The cell membrane can perform functions relating to, okay, so we have to select all that apply. So the cell membrane. So go ahead and review it. Okay, so it's a boundary. It regulates movement of substances in and out of the cell. It's many of the cell's actions that enable it to survive and interact with other cells use signal induction. So they detect cells from outside the cell and transmit them inward. And other molecules orchestrate the cell's response. The cell membrane also helps cells to attach to certain other cells. Okay, so let's see what they give us in the question here. So it detects signals. It controls the movement of cells entering the others, and it attaches to other cells. That's those 3. Good job. Nice. Okay, the prefix for above is
Be hyper.
hyper, correct? Good. In an aqueous water-based solution, if the phospholipids were unable to form a bilayer, they would form what? Okay. We haven't read about this yet. Okay, so we need to talk about if they were unable to form a bilayer. This is the cytoplasm. This is proteins. I think it's before this, when they were talking about the bilayer. Keep going. I think it's up here somewhere. A cell membrane, okay. The cell membrane's middle portion is oily. The cell membrane's middle portion is oily because it consists largely of the fatty acid tails of the phospholipid molecules. molecules that are soluble in liquids, such as oxygen and carbon dioxide, can pass through the phospholipid bilayer easily. However, the bilayer is impermeable to water-soluble molecules, which include amino acids, sugars, proteins, nucleic acids, and certain ions. Cholesterol molecules embedded in the cell's membrane interior help make the membrane less permeable to water-soluble substances. The relatively rigid structure of the cholesterol molecules stabilizes the cell membrane. A cell membrane includes a few types of lipid molecules, but many kinds of proteins. Proteins, specialized functions of the membrane. Membrane proteins are classified according to their positions in the phospholipid bilayer. Integral proteins extend through the lipid bilayer. Okay. Okay. Keep going. See if there's anything else about many ion channels. Okay, but that's not telling me the shapes that they. Is there anything above where we were at? So go up the page again. Is there anything I missed? Maybe here, hold on. Keep going. Water fearing and make, okay. Um, okay, we talked about the glycerol molecule. Make up the interior. The lipid molecules can move sideways, the stable film. Okay, but it doesn't tell me anything. Okay, keep going. It hasn't told me what they do if they can't. Let's see. Okay, that's cytoplasm, cytoskeleton. I don't know where this is. Go back to the question. Maybe I can find it from the a single layered structure with tails out and heads in. Oh, I don't know. I don't remember reading that at all. Hold on. Let me look it up and see what it says. Um, in an aqueous solution, if phospholipids, phos, let's see. They would form, let me see what it says. I don't know. It's not giving me. So phospholipids spontaneously form a bilayer, but if they couldn't, it says if they couldn't, but I don't know. I don't know what to say there. Hydrophilic heads facing out, and they're if the phospholipids have short tails, they may form spheres. If the tails are longer, they may form hollow. I'm not sure. I would say, yeah, I don't know the answer to that question. I would say B, a single layered ring structure with tails in, because they would put their tails in the cell. I don't know, but based on the fact that the heads like water, they would face on the outside. Let's see what it says. Yeah, because the tails don't like water, so they would try to be inside and they would stick their heads out, so they would form a ring with their tails in the middle of it. Yeah, there's no real reason for it. Okay, let's go on to, okay, hypo. Which one is it?
Below.
Right. Oh, good. Okay. What is cytosol? A type of vesicle, the liquid part of the cytoplasm, the membrane that separates the inside and outside of the cell, or the DNA in the nucleus. Cytosol. Okay. So let's see. The cytoplasm, I need to expand this. I can see it. The cytoplasm is the gel-like material that includes the cellular organelles. When viewed through a light microscope, cytoplasm usually appears as a clear jelly with specks scattered throughout. However, an electron microscope, which provides much greater magnification and the ability to distinguish fine detail, reveals that the cytoplasm contains vast and complex networks of migrans and organelles suspended in the more liquid cytosol. So cytosol is it's within the cytoplasm that that contains where the organelles and the membranes are. So let's see what the question says. I would say the liquid part of the cytoplasm would be cytosol, based on what we read. Yeah. It's the liquid that suspends everything. Cells are the smallest unit of life. We read that earlier. That's the smallest unit that you have. Okay. What is the function of ribosomes? They are where protein synthesis takes place. Those are the protein factories of our cells. Okay, what is A pointing at?
Would it be the cell membrane? I think the nucleus.
It's pointing at, I think it's pointing at the nucleus, yeah. Because the cell membrane would be on the outside. In muscle cells, blank aggregate forming microfills. So in muscle cells, what aggregates? I don't think we read this yet. So let's look down. Microfilaments and microtubules are types of thin, thread-like strands in the cytoplasm. They form the cytoskeleton and also were part of certain structures such as centrosomes, cilia, and flagella that have specialized activities. Microfilaments are tiny rods of the protein actin. They form meshworks or bundles and provide cell movement. In muscle cells, for example, microfilaments aggregate. So the answer to that is microfilaments aggregate, forming microfibrils, which help these cells contract. So microfilaments come together to form myofibrils. So microfilaments is the answer to that. Microfilament. Okay, next question. The term used to describe the cell membrane, because only certain substances can enter or leave the cell, is called permeable, selectively permeable, or impermeable. Which one? Okay, look at the blue word. A cell membrane is thin and flexible and composed mainly of lipids and proteins with a few carbohydrates. The cell membrane maintains the structural integrity of the cell and is selectively permeable, which means only certain substances can enter or leave. So selectively permeable. Okay. Good. The nucleus is the location of blank when the cell is not dividing. Okay, so there's my nucleus. And what's in it when the cell is not dividing? So that's the picture, but where's the nucleus, the nuclear membrane? The nuclear membrane consists of the phospholipid bilayers. It contains the nucleus and masses of chromatin. Okay. Nucleolus and chromatin. A nucleolus is a small, dense body composed largely of RNA and protein, has no surrounding membrane, and is surrounded by ribosomes form the nucleolus and then migrate. Chromatin consists of loosely coiled fibers of DNA and protein. When the cell begins to divide chromatin fibers coil tightly. They condense to form chromosomes. That's when the cell divides. But before they divide, go back up to before the picture. And let's see, the nucleus houses the genetic material which directs all cell activities. It is a large, roughly spherical structure enclosed in a double-layered nuclear envelope, which consists of inner and outer lipid bilayer membrane. Nuclear pores. Nuclear pores large enough to let out the RNA molecules, but not large enough to let out the DNA itself, which must remain in the nucleus to maintain the genetic information. I would say DNA. Go back to I would say DNA would be the one that, based on what we read there, because the vesicles are outside. All those other things are outside the nucleus in the cytoplasm. Okay, within the structure of the cell membranes, the phospholipid heads face blank. Okay, so the phospholipid heads face the okay, I have to read this weird. So the phospholipid heads are going to face outward and the tails are going to go inward toward the cytoplasm. So I would say the tails, I would say it's the second one. The phospholipid heads face the interior of the membrane, and the cytoplasms. Wait a minute, is that backwards? The phospholipid heads face. No, I think I'm right. The interior of the membrane, because the tails would face the cytoplasm, they would face the water. They don't like they face outward. I think I'm right. Let me see. I'm not sure which one is. Wait, let's see. Let's see if we're right. No, I got it backwards. So recall that the tails face each other. The phospholipid heads are in contact with the external environment of the cell. So, okay, so the tails face each other on the inside of the fluid. So, oh yeah, the tails face each other. So that would be the second part of the question. Okay, I got that wrong. The tails face each other. So basically, the phospholipid heads face the cytosol and the extracellular fluid. The tails face each other on the inside. The tails face each other in a lipid bilayer. The phospholipid heads are in contact with the external environment of the cell and the internal cytoplasm. Okay, that's why it's okay. It'll come back. It'll come back as a question. The liquid portion of cytoplasm is called cytosol. We had that question before. It's called cytosol. You can look it up if you need the spelling for it. See here? It's above cytoskeleton. So it says also includes protein rods and okay, let's see. Reveals that the cytoplasm contains vast and complex networks of membranes and organelles suspended in the more liquid cytosol. Cytosol. C-Y-T-S. Cytosol. Diffusion is a blank process which depends on concentration gradients. Okay, not sure what they want here. Diffusion is the tendency of molecules to form a liquid, solution, or air to move from means of higher concentration to means of lower concentration, but they're passive. See the word passive in red? They're passive mechanisms. So they're passive. I think that's the word they're looking for. It's passive. Diffusion is passive. A passive process. 2 S's. No. P-A-S-S. All right, there we go. There you go. Passive. Small spherical structures that, okay, do you remember the structures that drive protein synthesis?
Would it be cytoplasm?
It's in the cytoplasm, but what is the structure called? Starts with an R.
The ribosomes?
Yeah, that's correct. Ribosomes are structures that drive protein synthesis. Good. Nice. Okay. Diffusion describes molecules that do what? They move from high concentration to low concentration. So it's the first one. That's why it's passive, because they're moving from high to low. If they were moving from low to high, they would need energy. So that would be active if they did that. Okay. Microfilaments are tiny rods of the protein. I don't remember the name of it, but it's muscle protein, right? So I need to read about microfilaments again. Okay, microfilaments are tiny rods of the protein. What's the name of the protein?
Actin?
Actin. That's what they want. Actin. Good. Okay. Oxygen is at a higher concentration outside the cell compared to the inside of the cell. and the cell membrane is permeable to oxygen. Based on the laws of diffusion, which way will the oxygen molecules move? Are they going to move from outside to inside or from inside to outside? If it's at a higher concentration outside, which way is it going to move?
Would be inside to outside.
Diffusion always moves from where it's higher to where it's lower. Where is the oxygen higher?
Outside.
So it's going to move from the outside to the inside. That's why they had to tell you that. If it's diffusion. Now, if it's active transport, then it's different. So yeah, okay. The blank is a relatively large, usually spherical structure that contains the genetic material which directs the activities of the cell. Where's that big structure that looks like a ball? What do we call that?
The nucleus.
The nucleus, right? Mm-hmm. Good. Nice. Simple blank excuse me, let me read that again. Simple blank occurs due to the random movement of atoms, molecules, or ions in air or liquid from higher concentration to areas of lower concentration. What do we call that movement again? Diffusion, also called simple diffusion, is the tendency of molecules or ions in a liquid solution or air to move from regions of higher concentration to regions of lower concentration. Diffusion, the word in blue. Diffusion. Good. In an aqueous solution, okay, we did this one before. In an aqueous solution, water-based, if the phospholipids were unable to form a bilayer, they would form a single layered structure with tails in and heads out. Because they want the tails to face inward, where they don't have the water, because they don't like water. Remember? In comparing two solutions with the same volume, the one with more solutes has blank water than the one with fewer solutes. Not sure we've read this yet. What do you think? You want to read it? Let's read it. Okay. The difference in, okay. Go up to, no, I mean, go back up because I need to read. Okay. Um, Diffusion occurs because molecules and ions are in constant motion. Each particle moves at random in a separate path along a straight line until it collides and bounces off another particle, changing direction, then colliding and changing direction once more. Molecules and ions exhibit a universal quality where they seek to reduce collisions and spread out as much as possible. You might say they want personal space. This is true in living and non-living organisms. Collisions are less likely if a solution has fewer particles. So there's a net movement of particles from a region of higher concentration to a region of lower concentration. The difference in concentration of particles from high to low establishes what is called a concentration gradient. Diffusion occurs down a concentration gradient, which is beneficial for cells because it is passive. That is, it requires no energy. Keep going. We haven't gotten to the solute solvents yet. With time, the concentration of a given substance becomes uniform throughout a solution. This state is called diffusional equilibrium. Random movements continue, but there is no further net movement, and the concentration of a substance remains uniform throughout the solution. Okay. Then it talks about the diffusion of sugar through water. Keep going. So go up to below the picture. Up. Go up the. Okay, so it says a drawing dissolving sugar cubes. A sugar cube can dissolve and diffuse from regions where they're more concentrated. Okay, I need solute and solvent, but let's go up a little bit. A cube of sugar, a solute, put into a glass of water, a solvent, illustrates diffusion. At first, the sugar remains highly concentrated at the bottom of the glass. Diffusion moves the sugar molecules from the area of high concentration and disperses them into a solution among the moving water molecules. Eventually, the sugar molecules become uniformly distributed. Okay. Let's see what the question says again. The one with more solutes, the one with more solutes I'm not sure, but I would say if it has more solutes, it has more stuff in it. Um, I don't think the volume changes. But let's keep reading because we haven't read anything about volume yet. But I don't think the volume changes. I think the concentration changes, but not the volume. That's what I think. Go ahead, let's keep reading it and see if it has. Okay, I read that. Keep going. Keep going. Keep going. More. Okay. Cell membrane. Diffusion of a substance across a cell membrane can happen only if the cell membrane is permeable to that substance and the substance is small and without an electrical charge, and a concentration gradient exists such that the substance is at a higher concentration on one side of the cell membrane or the other. Yeah, but it doesn't say anything about volume of it. Keep going. I'm gonna say, okay, go to the question. I'm gonna say that it has, I'm not sure, but I think if you had more stuff dissolved in it, it would be more concentrated. But I'm not sure if the volume would be the same because I'm not sure if the volume refers to everything or the water. Try more and see what you get. I'm not sure about this. No, it has less because um because there's more stuff in it. So the one that has more stuff suspended in it, the water is diffusing, so it would have less water in it than the one with fewer solutes. Okay, go to the next question. The process that occurs due to the random movement of atom molecules, that's diffusion. Diffusion again. They're asking you about diffusion in a different way. Okay. Okay, what is what is the term given to the difference in concentration of particles from one area to another? The difference in concentration is the gradient. The movement is the diffusion, but the difference is the gradient there. Okay, next is when molecules move down a concentration gradient, it means that there is a movement of particles from a region of lower concentration to higher concentration. Is that how it works? True or false? Okay, so they move from, look, higher concentration to lower concentration. See on the second line? Higher concentration to lower concentration. So that's false. Nice. When molecules move down, okay, they just asked you this question. When molecules move down a concentration gradient, it means that there is a net movement of particles from a region of lower concentration to higher concentration. That's false. They asked you the same question twice. I don't know why they did that, but the process of diffusion is possible in air or in solution, air only or solution only. It's possible in air and in solution. It's possible in both. You can diffuse through air. Fragrances do that. Perfume does that. The goal of diffusion is to attain equal concentration of solute throughout the solution. This is known as what? when you have, okay, go ahead. Okay, let's keep going. Okay, I think go up. I don't think we read the last paragraph. So it says, let's see. That is, it requires no energy. With time, I'm reading toward the middle of that last paragraph. With time, the concentration of a given substance becomes uniform throughout a solution. Uniform means the same. This state is called diffusional equilibrium. Random movements continue, but there's no further net movement, and the concentration of a substance remains uniform throughout the solution. So equilibrium is your answer there. Equilibrium. Yes. The movement of molecules in solution to naturally disperse is diffusion. The natural disbursement of molecules, or maybe it's active transport now that I think about it, but I think it might be check and read it and see. But I think it's okay. Okay, keep going. Keep going. Diffusion across a cell membrane. Okay. Equilibrium is never reached. Extracellular oxygen is maintained. This concentration grid always allows. Okay. Okay, keep going. I think it's probably, they haven't talked to me about active transport yet, have they? I don't see anything about, go to the question again. To naturally disperse. No, if they're naturally dispersing, they're passive. That would be diffusion. I'm pretty sure that's diffusion. I'm not sure why they're asking you that in so many ways, but I think that's what they mean. Yeah, because active transport would require energy, and that wouldn't be natural. You have to introduce it to it. Okay, next question. If a solute can cross a cell membrane and the solute has a concentration gradient, so provided that you have a solute like water that can pass and the solute has a concentration gradient, it's going to form a solute can cross a cell membrane. Not sure what it wants here, but whether it wants diffusion or endocytosis, because it would go into it, basically. That's what it would do. Try diffusion and see, but diffusion is usually water across a membrane. So I'm not sure if it wants that or not. Go ahead and it might be, okay, yeah. Because it would flow into the cell, but diffusion is the passage of substances from greater to lesser concentration. So, okay, next question. Solution A has a higher concentration of salt than solution B. Therefore, solution B has a blank concentration of water than A. Okay. So basically, A contains their saltwater solutions. A contains more salt than B does. So B would have a higher concentration of water than A does. I think. B would have a higher concentration of water because B has less salt, is what it's saying. If A has more salt, it has less water. If B has less salt, it has more water. So it would be a higher concentration of water than A would have. If it asked me about salt, that would be different. The term concentration blank refers to the difference in concentration between, that's concentration gradient. Gradient is the word there. Okay. Okay, next question. This blood gas moves across cells by, okay, we have to look this one up. This blood gas moves across cells by simple diffusion. Read about the concept. So we have to read what moves. Okay, we read that we need blood gases. So we need to go down to where it talks about blood. Keep going. This is about, we read that. We read about the sugar. Keep going. Keep going. Okay, so here it says, consider oxygen and carbon dioxide, which are soluble in the lipids that form much of the cell membrane. Oxygen diffuses into cells and carbon dioxide diffuses out of cells, but equilibrium is never reached. So they both diffuse, oxygen and carbon dioxide. One moves in and the other moves out. It didn't specify which one. Both diffuse. Moving down a concentration gradient is the movement from, remember, it moves from an area of high concentration to low concentration. So that's the last answer on your list. High concentration to low concentration. As opposed to simple diffusion, substances which are larger may require special membrane proteins for the process of blank diffusion. Okay, we haven't gotten to this yet, but this is the more active one. So now we've talked about passive. We've talked about the passive one, but keep going. This is passive, but keep going. Keep going. Okay, so substances that are too large, such as a glucose molecule, or that have an electrical charge, are not able to pass through the lipid layer of a cell membrane. To move down the concentration gradient, they need the help of a membrane protein. This process is called facilitated diffusion. It is also referred to as facilitated transport, and as with simple diffusion, does not require an expenditure of energy. Facilitated diffusion is the answer to that question. Facilitated. Nice. Diffusional blank occurs when the concentration of a given substance becomes uniform throughout a solution and there is no further net movement of molecules through the solution. What do we call that? When it becomes the same.
I think it's gradient.
It's moving down the gradient, and when it becomes uniform, when it becomes the same, it's equilibrium. Because equal means the same. Diffusional equilibrium occurs when the concentration of a given substance becomes uniform throughout. Because it doesn't move anymore because they're equal. It's moved down the gradient already. And the state that we get is called equilibrium. We do that. Active mechanisms do or do not require energy. Active mechanisms.
I think they do require.
They do require it, right? Passive ones do not, but active ones do. Good job. Next. Diffusion can occur where the substance has an electrical charge. There exists a concentration gradient across the membrane. Okay. That's right. And the membrane is permeable to that substance. The third one. It has to be permeable to it in order to get across it. If it's not, it's not gonna fit. It's not gonna make it. Very good. Active transport explicitly requires the use of, okay, I don't know if they want energy or some other word. So let's look at the okay. Two processes use cellular energy to move substances into or out of a cell without actually crossing the cell membrane. I think, okay, go up a little bit. I don't think we're, no, up the page. I think keep going because it was talking about carrier molecules. So let's keep going. Okay, active transport is similar to facilitated diffusion in that it uses specific carrier molecules in cell membranes. Okay, specific carrier molecules in cell membranes. It differs from facilitated diffusion in that particles are moved from regions of lower concentration to regions of high concentration, and energy from ATP is required. Okay, so go to the question. It's I would say energy because it mentions membrane carriers. So try energy there and see if that's what they want. That's a little bit hard to tell. Okay, yeah. Because they mentioned membrane carriers. I thought maybe they wanted carrier proteins, but it mentions carriers in the sentence, ATP or energy. Okay. They would have taken ATP or energy. Okay, this one I missed before. Let's see if I can get it now. In comparing two solutions of the same volume, the one with more solutes has, okay, so if it has more solutes, then it has less water. It's less concentrated. It has less water than the one with fewer. Because I missed that question earlier. Okay. The exchange of oxygen and carbon dioxide in the lungs is an example of simple diffusion. They are simply diffused. We read that earlier. Oxygen goes in, carbon dioxide goes out. That's a diffusion mechanism. Both simple diffusion and facilitated diffusion are passive molecules, passive transport mechanisms that move molecules down a concentration gradient. Simple diffusion is passive, facilitated diffusion is passive. So they're both passive and they move it down a gradient. So the first question, the first answer there. The energy from ATP is used to power what? So when we need ATP or we need energy, you can read about it if you need it. We just answered a question about this. Okay, so active transport is similar to facilitated diffusion in that it uses specific carrier molecules. It differs in that particles are moved from areas of low concentration to high concentration, and energy from ATP is required. That's active transport. That's active transport. Because remember, facilitated diffusion is passive. So, okay, if you're moving against the concentration gradient, you'd be going from low concentration to high concentration. So you'd need to push against it. That would be active transport. In active transport, substances are moved against the concentration gradient. Active transport. Good job. Nice. Okay, we are at about an hour and 33, so I can give you about 25 more minutes. Okay? Okay, let's see what we've got here. Oh, okay, more of the same. Which root word means without? Not sure if it's Anne or Anna here, but let's read about it and see. Okay, Anne. A-N is without, Anna is up. Oh, okay, I didn't know that. Learned that one. And, so Anne is the answer there. Wow, there's 45 concepts in here. No wonder you wanted to start it. What is the function of enzymes? To control the rate of metabolic reactions. They speed them up. They speed up the rate of. Okay. The type of reactions that require the addition of energy are catabolic, I believe, because they're the ones that, I'll tell you why. Go ahead and click on it, catabolic, and I'll tell you how I remember it. Oh, wait. Oh, that's wrong. I didn't know that. Anabolism, the building of molecules, requires energy, while catabolism release. I thought it was reversed. I thought that catabolic reactions. Okay. Have to read about that more. We'll come back and read about that one. Because I thought that anabolic reactions they broke down things. Okay. The type of chemical reaction which, okay, I don't know this one at all. The type of chemical reaction that releases anabolism provides the biochemicals required for cell growth and repair. A type of anabolic reaction called dehydration synthesis joins many simple sugar molecules to form larger molecules of glycogen. When monosaccharides join an OH hydroxyl group from one monosaccharide molecule and a hydrogen atom, they form an OH group of another. Okay, is that? Okay, so what is the question again? Oh, it would be dehydration synthesis. So you have to write dehydration in the box. Dehydration synthesis. All right. Breaking a peptide bond requires the addition of a what kind of molecule? I don't know this one either. Breaking a peptide bond requires the addition of what? You have to read it and find out. Okay, catabolic reactions break down larger molecules into smaller ones. An example of a catabolic reaction is hydrolysis, which breaks down carbohydrates, lipids, proteins, and splits water molecules in the process. Okay, but this isn't talking about peptide bonds. Let's see. In this case, the bond, hold on. In this case, scroll down. In this case, the bond between the simple sugars that form sucrose breaks. Hydrolysis is responsible for digestion. Okay, keep going. Okay, keep going. To break a peptide bond. I'm not sure what that. Let's go to the question and see what. Yeah, it doesn't tell me that. Hold on. I have to look it up. Breaking the peptide bond requires. Okay, so. Okay, hold on. Look it up. Okay, let's see. Breaking a peptide bond. Oh, I guess that's what happens in hydrolysis. Requires the addition of. Okay, let's see. There's a flashcard here. Let's see if it tells me. Well, we have that question. That's enzymes. Water molecule. It's a water molecule. Because it's hydrolysis. It's when you break apart, you add water molecules to it. Okay. Let's continue. Which word includes the stem for without? Without. Well, what is the stem for without? Let's review it. What is the stem for without? So the stem for without is an. So we would need a word with an an on it. So let's look at the words. So it would be anaerobic without air, and yeah, I think it's anaerobic. Because anabolism is ana. The reason that it's not anabolism is because that bolism by itself is not a word, but aerobic by itself is a word. It's just a different spelling. Anaerobic means without oxygen. So, yeah. Next question. Enzymes are considered catalysts because they, okay, why are they considered catalysts? Let's look. Okay, so enzymes make these reactions possible by lowering the amount of energy called the activation energy required to start reactions. In this way, enzymes speed metabolic reactions. This acceleration is called catalysis, and an enzyme is called a catalyst. So they are considered catalysts because they metabolic reactions. Okay. Enzymes make these processes possible. So I would say they speed metabolic because they speed metabolic reactions, which is called catalysis. So they increase the rate of metabolic reactions. Okay. Enzymes are blank that control enzymes are proteins that control the chemical reactions of cells. I think they're proteins. Let's review it, but I think they're proteins. Okay, so special proteins, yeah, they're proteins. Special proteins called enzymes are vital to all of these activities because they allow chemical reactions in the body to proceed fast enough to sustain life. So yeah, they're proteins. Yes. Okay. Why are enzymes able to function repeatedly, catalyzing multiple reactions? They are not consumed during the reaction. They act as both substrate, reactant, and product, or they become part of the product. They can function repeatedly. Why? Well, because it says enzyme molecules are not consumed in the reactions they catalyze and can function repeatedly. So they don't get used up, remember? Enzymes can function repeatedly because they don't get used up in their reactions. So go to the question. They are not consumed during. Okay, next question. A major distinction between catabolism and anabolism is the temperature at which the reaction occurs, the requirement for an energy output, and the size of molecules included in the reaction. Which one of those? Okay, let's look. The reactions of anabolism synthesize or build larger molecules from smaller ones, requiring an input of energy. The reactions of catabolism decompose or break down larger molecules into smaller ones, releasing energy. These reactions are often coupled together, where the energy released from a catabolic reaction is used to fuel an anabolic reaction. Okay. Okay, so one releases energy, one requires that you put energy into it. So the requirements for an energy input. Yes. In joining two molecules by dehydration synthesis, a blank group is removed from one molecule and a blank group from the other. A new chemical bond then forms from the molecules. So you have to select the words that apply. In dehydration synthesis, so we have to look at dehydration synthesis again and see what it says. So anabolism provides the biochemicals needed or required for cell growth and repair. A type of anabolic reaction, so that means it requires energy input, right? Called dehydration synthesis, joins many simple sugar molecules to form larger molecules of glycogen. So you have when monosaccharides join a hydroxyl group, that's OH, from one monosaccharide molecule, and a hydrogen atom from a hydroxyl group of another molecule are removed. As the hydrogen and hydroxyl react to produce a water molecule, the monosaccharides are joined as a shared atom. So it's hydrogen and hydroxyl, I think. Hydrogen H and a hydroxyl, I believe so. Yeah. Enzymes usually bind to, I don't know. Okay, each type of enzyme is specific, acting only on a particular molecule called a substrate. Many enzymes are named after their substrates with "ase" as a suffix. So it's a specific substrate is the answers to that question. A single type of molecule. The type of catabolic reaction called blank results in the breakdown of protein or polysaccharide molecules. Water molecules are split in the process. I think that's hydrolysis, but let's review it. I think it's hydrolysis. Yeah, an example of catabolic reaction is hydrolysis, which breaks down carbohydrates, lipids, and proteins and splits a water molecule. So hydrolysis, the green word or the blue word there. Hydrolysis. Okay. Sequences of reactions controlled by enzymes as part of metabolism are called metabolic pathways, protein folding, endocytosis, or cellular transport. Okay, specific enzymes catalyze each of the hundreds of different chemical reactions that constitute cellular metabolism. Sequences of enzyme-controlled reactions called metabolic pathways lead to synthesis or breakdown of particular biochemicals. So, metabolic pathways is your answer there. A blank is considered a catalyst. Okay, what do we call it? The thing that accelerates chemical reactions.
Would it be synthesis?
That's putting it together. What do we call the protein that accelerates? It's an enzyme. An enzyme is considered a catalyst because it accelerates chemical reactions. An enzyme. That's right. Yes. Radiation, heat, and some chemicals can cause a protein to unfold called what? Okay, so almost all enzymes are proteins. Like all the proteins, exposure to heat, radiation, electricity, certain chemicals, or fluids with extreme pH values can denature or otherwise inactivate enzymes. So denature or denaturation. Let's look at the question called denature. Yeah. I think that's try it. Denaturation or okay, they needed the noun, so they needed denaturation. This is the verb. That's why you got it wrong. And that was my fault. Denaturation is the noun or denaturing. Okay. Denature is the action of doing it. So you had the right word, but the wrong form of it. And that was my fault. Okay, but it's the right term, it's just the wrong form. Enzymes participate in metabolic reactions and generally increase the rate of reactions. So the second one and the third one, they increase the rate of reactions and they're not consumed in the process of doing it. Yeah, there you go. Exposure to, okay, find the things that can denature enzymes. Exposure to what? There's 3 of them on here.
I think would be heat.
Heat is one, extreme pH is another one, and radiation is another one. There are more, but those are the main 3. Yeah. Okay, so let's the type of reactions that require extra energy are anabolic. I missed this one before, remember? So it came back to you. I think I need to stop here,
Okay.
Daniela, but tomorrow, 9 o'clock, you said?
Yes.
Okay, I'll see you then. I sent you a receipt for January, okay?
Okay.
We didn't have a full month, but I sent you a copy and I already sent it to Clockwork. And I sent a copy to Karina as well, but she's out of the office, I think, until Monday. All right.
Okay.
We'll talk. Keep up the good work. I miss seeing you.
Yeah, me too.
Take care, okay?
Okay, thank you.
Bye.
Bye.